Hi there, this is Dr. Rosario. This is our first video in our module on microbial growth. In this video, I'm going to define for you what we mean by microbial growth. We're going to talk about some of the important aspects of the growth of bacteria in pure cultures. And then we're going to also talk about how we measure microbial growth. And then finally, I'm going to talk about how microorganisms grow in association with each other. And we're going to discuss the importance of biofilms. When you think about growth, you probably think in terms of growth of animals or growth of plants, right? Where an organism begins as a very small organism in infancy and grows into adulthood or into maturity. For microorganisms, and bacteria particularly, we look at growth in terms of population size, not in terms of an increase in size. So microbial growth starts maybe with a single organism, but that single organism will divide to produce many different organisms. And that in turn produces what we call a colony, right? Remember, a colony is a group of microorganisms that all came from a single parent cell. They are genetically identical to one another. And we can see them growing when we look at solid media in the laboratory. We can see our colonies on the surface of our agar plates. For multicellular organisms, they grow through mitosis, through mitotic cell division and the addition of cells. For our prokaryotic organisms, they grow in population size through binary fission, where you have a single cell with its single chromosome, and that chromosome is replicated, and then ultimately the duplicated chromosomes are divided into two daughter cells. This cycle of DNA replication followed by cell separation provides the basis for bacterial growth. Generation time is the amount of time it takes for the population to double using binary fission. This is really important because for some of our bacteria, that generation time can be very short. For example, for E. coli, the generation time can be less than 20 minutes under optimal growth conditions. So you can have the doubling of a population every 20 minutes or so. That can take you from just a few microorganisms to a massive amount of microorganisms in a very short period of time. This doubling of microorganisms with each generation is known as exponential growth. So here we start with one cell, and that one cell becomes two cells. Those two cells become four cells. Those four cells become 8 cells, then 16, 32, and 64, all in a very short period of time. Within just 6 generations, we went from 1 cell to 64. We can look at these numbers in terms of powers of 2. So 1 is 2 raised to the power of 0. 2 is 2 raised to the power of 1. 4 is 2 squared. 8 is 2 cubed, and so on and so forth. So for each generation, we're increasing exponentially by a power of 1. So let's take a look at what that would look like if we continue this out. And I'm going to show you a graph of that data. So here you can see after 20 generations, we started with 1 cell, and now we have over 500,000 cells. Now, when we look at this, it doesn't look like there's much change in the earlier generations because those numbers, as you get to the later generations, are so big. So we need to look at this a slightly different way. We can graph this on a logarithmic scale, and that gives us a nice linear slope that then we can use to calculate our generation time. So that line is directly proportional to the rate of growth, or the amount of time it takes for each generation to double. This becomes important when you're wanting to compare growth between two different populations. We're not going to do that in this class, but it is something just to keep in mind that oftentimes when we plot the growth of microorganisms, we do so on a logarithmic scale, multiples of 10 rather than just the raw numbers. So keep in mind when we're talking about the growth of a bacterial population, we're usually talking about the growth of bacteria in the laboratory, in what we call batch culture. So that's when we have our bacteria growing in a test tube in broth culture, right? And you have a pure culture of just a single species of microorganisms. And when you start that population, right, with just a few cells, initially when you put those cells into that fresh media, into that fresh broth, it takes them a little bit of time to adjust to that environment. That period of time where those cells are adjusting to their new home, they're changing their gene expression in response to their new environment. They're taking up nutrients. They might be building some biological molecules. That period of time is what we call the lag phase. At that point, there's no increase in the number of bacterial cells present in that growth medium, but instead they're in that adjustment phase. So that's called lag phase. Eventually, those cells will start to replicate. And here, this graph is on a logarithmic scale, and you can see that exponential growth begins, and we call that exponential phase log phase, right? At that point, your cells are replicating in that batch culture at a constant rate. They're doubling according to that generation time. Eventually, in that test tube, what will happen is waste products will start to build up, nutrients start to become depleted, and we hit what's called stationary phase. In stationary phase, there's a plateau in the total number of living bacterial cells. Some of the cells are starting to die off because of the waste products building up. Some are still dividing, but the amount of cell division and cell death is relatively equivalent. So now we're in what's known as stationary phase. What then can happen is that as that culture gets older and older, the cells may enter into what's called death phase or decline phase, where the entire culture starts dying off. Some bacteria can persist in that stationary phase for longer periods of time, where that death phase doesn't really happen for quite some time. It could be days or weeks. But some bacteria in a test tube may die off real quickly if you don't transfer them to fresh growth media. So they'll enter into that death phase. So this growth curve that we see is characteristic, again, of bacteria growing in the laboratory, right? In media where we're supplying the nutrients and it's a closed environment. We can also grow bacteria in continuous culture in what's called a chemostat. So in a chemostat, you're maintaining bacteria growing at that exponential rate where the cells are doubling constantly. And the way that you're able to maintain that continuous culture, that constant cell doubling, is through adding nutrition and then removing waste products, and even sometimes removing cells from that population. We use this continuous culture process called a chemostat, oftentimes in industrial production of bacterial products, or even sometimes in food culture, you'll use a chemostat. It's not something that we're going to use in our laboratory. But again, a chemostat is used for continuous culture. The key here is that it's not a closed system. There's an input of nutrients and there's an output of waste products and even sometimes cells. You have a continuous supply of nutrients. We're removing the waste, and we have a constant log phase, right? The cells are constantly doubling. That's what occurs in continuous culture. That's going to be different than what we have in our batch culture in our test tubes. Okay, so if we're growing microorganisms, we want to have ways to measure how quickly they're growing, right? To count the number of microorganisms we have in our cultures. There are a variety of different methods that we can use. We have what are known as direct methods, where we're actually counting the microorganisms directly, or we have indirect methods where we are measuring products of microbial growth or consequences of microbial growth. So our first direct method is known as a direct microscopic count, and it's exactly the way it sounds. Here you take microorganisms out of your culture, you take a sample out of your culture, and you put it on a glass slide. And that glass slide has a grid etched in it. And then you can see that grid under the microscope along with your bacterial cells. Usually you use some sort of staining technique to be able to visualize the cells. And then you can count the number of cells and then count up the number of cells per unit volume on that little glass slide. So this little glass slide is called a hemocytometer. These are used to count bacteria. They're also used to count other types of cells as well, including blood cells and tissue culture cells for eukaryotes. So the problem with the direct microscopic count is that it's very slow and tedious. You can imagine we're talking about very large numbers of cells and sitting there over a microscope and counting each individual cell that you see. That takes a lot of time. The other important thing to keep in mind is that when you count the cells, you're not distinguishing between those that are live and dead, right? So if you are trying to measure microbial growth and you want to know, are these cells in exponential phase where everybody's growing and replicating, or are they in stationary phase where some are dead and some are alive? You want to differentiate between live and dead cells. We have a way around that. So we can use a fluorescent stain that will allow you to differentiate between live and dead cells. So what's happening in this particular image is this stain. You have a green stain that is able to penetrate the cells that are only alive, right? The dye is only able to get into cells that are living. And then you have a red stain that can get into cells that are dead, where their membranes have been disrupted in some way. So that when you're looking at this, the ones that are green are those cells that are alive, but the ones that are showing up as red are the cells that are dead. And you can put this on a hemocytometer as well, so that you can count live versus dead cells. So this is also a direct microscopic count. But here we have that ability to differentiate between live and dead cells. This is going to require a little bit more specialized equipment. For example, you need a fluorescent microscope rather than just a standard light microscope. Again, slow and tedious, and you've got that staining process that you have to do on top of the counting. Another way that we can count cells directly is through using something called a Coulter counter. This is an electronic device that will actually count cells as they pass through the chambers of an electrode. And then you can get a direct count of the number of cells that are present in a solution. But like the direct microscopic count, you can't tell the difference between living and dead cells using a Coulter counter. One way that we can differentiate between living and dead cells is by using what's known as a viable plate count. So on a viable plate count, you have to remember that each colony, each single isolated colony you see on a plate represents a single viable cell that landed on that media and started to divide. In order to be able to count those colonies, you have to have just the right number of cells distributed on the surface of that media. So for the viable plate counts for your standard Petri dish agar growth, we're looking at a countable range of about 30 to 300 colonies. If you have less than that, there's too much statistical variation. If you have more than that, the colonies are too close together in order to count them. So we call that too numerous to count. So to use a viable plate count, you have to get a specimen that has just the right number of cells within it to then place into your culture media. There are a couple of different ways that we can generate plates for counting. So one is called the spread plate method, and this is probably the simplest to understand. Here, you take your specimen of bacteria, you take a small volume of that specimen, and you spread it on the surface of your agar, of your growth media. And then you incubate it, and then the colonies that come up, you count. An alternate method would be called the pour plate method, where here you take molten agar, you mix your specimen into that molten agar, and then you pour that agar onto your Petri dish. You let it spread out, and then you incubate it and allow for the colonies to grow. In this situation, what happens, your bacteria grow on the surface, but they can also grow within the agar itself. So you get those that maybe are not able to grow right there on the surface in the high oxygen environment. So here, the bacteria are mixed with molten agar. So those are two different ways to generate a viable plate count, the spread plate or the pour plate. In both cases, you need to have a specimen that has just the right number of cells in it. So here I have a test tube, and this test tube has E. coli in it. That E. coli is grown in rich media, and it's pictured at late log phase, so it's nice and cloudy. That specimen can have more than 1.5 billion cells per milliliter. That's a lot of zeros, right? 1 billion has 9 zeros behind it. So if I was to take 1 milliliter of that culture and spread it on a plate, I would end up with 1.5 billion colonies. That would be too many to count for sure. So in order to get a plate that has colonies in the countable range, we have to do what's called a dilution series, where you take your initial specimen, you add that specimen to test tubes with a set volume of usually growth media, or it could be water, depending on what you're working with. And you do what's called a serial dilution, where you dilute out your culture so that you get a small enough amount per unit volume, a dilute enough amount of cells per milliliter, so that you can take a portion of that specimen and spread it on your plate and get it into the countable range. So we use a dilution series when you start with a specimen that has too many bacteria to count. On the other hand, sometimes you're working with very dilute specimens where there's very few bacteria in that sample. And if you want to be able to count those, you have to use an alternative strategy. So an example of this would be in water, right? In a freshwater environment where you're trying to sample to measure the number of bacteria in that freshwater sample, the bacteria may be very dilute. And instead what you do is you concentrate the bacteria by running a large sample of water through a filter. The filter catches the bacteria on its surface. So in this case, the filter usually has a pore size of about 0.5 micron or 0.5 microns. Remember, most bacteria are about 2 microns or larger. So you catch the bacteria on the surface of the filter, and then you take that filter out of the cartridge and you place it on top of some growth media. And in this picture, the growth media that they're using is a differential media that allows for your fecal coliforms, right? Your E. coli and organisms like it that grow in uh, animals' intestines, right? Those fecal coliforms, those poop-associated bacteria. It's differentiating those, right? Those fecal coliforms in this example are showing up as blue. So we can count the number of fecal coliforms in that water. So this process of membrane filtration is going to be used for dilute specimens, right? Like water samples. We see this used a lot in water treatment facilities. So all of the methods that I've talked about so far are considered direct methods of counting microorganisms. A couple of our indirect methods, one is called turbidity. So this machine that is in this picture is called a spectrophotometer, and a spectrophotometer measures the amount of light that passes through a specimen. So here we're measuring what we call turbidity of a culture specimen as a way to measure microbial growth. It's going to be an indirect measure, right? Because we're not measuring the number of bacterial cells. Instead, we're measuring the scattering of light. So inside this spectrophotometer, you have a light source. That light source emits light within the visible spectrum. And then you have a filter usually that is included that will choose a particular wavelength of light. Most of the time we use 600 nanometers. That's right in the middle of our visible range. And that light is passed through your specimen. And then on the other side of your specimen, there's a detector. So in this picture, we have the detector is just that blue box. That detector is measuring how much of the light that was emitted by that light bulb is actually passing through our specimen. In this example, we have just growth media alone, right? So it's clear, and most of the light is passing through the specimen. But if you look at this particular picture, you have a culture that has lots of bacterial cells in it. So that light is being scattered by those bacterial cells, and you get less light passing through. So basically what turbidity is it's measuring the amount of cloudiness in your liquid culture, in your broth cultures. Just like your direct microscopic count, we cannot differentiate between live and dead cells. We can just measure kind of the total amount of cloudiness or the total amount of growth in your culture. This is a rough estimate of microbial growth. It's not a direct count, so we can't tell the exact number of cells that are in our specimen, but it is very quick, and you can do it in a very short period of time. It doesn't require much specialized training or specialized equipment, so it's very easy to do. This takes a whole lot less time than a direct microscopic count. Another indirect method that's a little bit more high-tech that I wanted to mention is something called real-time PCR. And we're seeing real-time PCR being used a lot more in the clinical lab for detection of pathogens and for quantification of pathogens. So what you do with real-time PCR, and I've got some pictures on the slide for you of this fancy machine and these plots that the machine spits out at you, but essentially what it's doing is it is counting or quantifying the amount of DNA or RNA in our sample, all right? So the more bacteria you have, the more DNA or RNA you have present in that specimen. And we can use probes that can detect specific types of bacteria. So this is why it's really useful in the clinical environment. One of the tests that was used for COVID-19 was a real-time PCR-based test. where they would measure the amount of viral RNA in a sample as a way to measure to determine whether or not a person actually had COVID-19. And then they could also measure how much virus they had in their system. So this is going to require a lot more technical skill, and this equipment is incredibly expensive. So here you're getting much more accurate, more precise measurements, not counting individual cells, right? So this is more of an indirect method, but it can be highly precise.